today. We have Ronan Carroll. He's just been in our department for a couple years, Department of Biological Sciences, and this is his thing. Before I bring him out here, I want to say, yeah, we had to reschedule this because he had conflicts and he did want to be as close to St. Patrick's Day as possible. And he has been able to bring, his wife is here, his mother-in-law is here, and I even have a representation of his dad, because he is Irish. Okay. So he was born in Sligo, Ireland, and he did his initial research in Trinity College in Ireland. He's done a bunch of postdocs, and he has studied all kinds of nasty bugs in Salmonella, Strep, Shigella, Mrsa, and this last one I had to have him tell me about, Bartonella, which is cat scratch fever. So he's like studied it all, and he's going to share some of his knowledge with us. Ronan.
Thank you, Janet, for the introduction. Can everybody hear okay? And thanks, Dad, for coming. I didn't know you were going to make it. So, yeah, thanks for the introduction. And so the title that I'm going to talk about today, The Rise of the Superbugs, and I kind of threw in this subtitle just before I came here, Why We Should Be Afraid and What We Can Do to Help. And so that's what I'm going to try and convey to you guys today. We'll talk a little bit about the rise of these superbugs, sort of what's driving that. And then towards the end of the talk, I would like to talk to you a little bit about stuff that we can do as general members of the public to try and combat this. But I also want to talk to you a little bit in brief details about some of the research that's ongoing in my lab right now to try and address this topic and to try and address these superbugs and things that we can do to combat them. Well, Janet kind of stole the thunder from my first slide here because as you guys have probably heard from the brief opening that, yeah, he has an accent. And so the first question most people kind of runs through people's mind is this, which one is it, right? It's either Scottish or Irish. You get a 50-50 shot, but Janet's already given it away, and so I am from Ireland. And so the timing for today really couldn't be any better, right, because tomorrow's St. Patrick's Day. And it's always really fun being an Irish person in America on St. Patrick's Day. But there's a couple of things that I kind of want to address, a couple of issues about St. Patrick's Day in America that I kind of just want to clear and clarify before we go any further. So if you just give me a few minutes before I get stuck into the superbugs, right? And the first thing is this. I want to introduce you all to the symbol of Ireland. The symbol of Ireland is the shamrock, or as we would say in Gaelic, it's a shamroig. And the shamrock, as you can see here, has 3 leaves. Okay, it's got these 3 heart-shaped leaves, and it is the symbol of Ireland. And you see it everywhere in Ireland. You see it on national monuments. You even see it on our national airline. So this is Aer Lingus, and you can see here on the tail fin we have this shamrock. Shamrock is not to be confused with a clover. A clover is a 4-leaf. You can see 4 leaves here, and it's a symbol of good luck. Now, the reason I bring this up is because I want you guys tomorrow, when you're walking around town, and we see a lot of sober people walking around town tomorrow, wearing their St. Patrick's Day t-shirts, I want you to look out and see how many clovers you see and how many shamrocks you see. Right? Just watch out for that because it's the shamrock, the 3 leaves, that is the symbol of Ireland, not the 4 leaves. 7 leaves is a completely different story. And I'm not going to talk about that today, but that, some people might say that's the symbol of really, really good luck, but we're not going to talk about 7 leaves today. Okay, so as I said, tomorrow is March 17th, it's St. Patrick's Day. And it's one of the interesting things about being an Irish person living in America is, you know, for 364 days out of the year, people love hearing your accent. They think it's hilarious. But on St. Patrick's Day, they all think your accent is fake. And so every March 17th, people are like, come on, cut it out. It's not funny anymore, right? But I am from Ireland. This accent is real. But one of the things that really bugs me about this country on St. Patrick's Day is the big debate of, is it Paddy's Day or is it Patty's Day, right? And let me just clear this one up right now. It's Paddy. It's not Patty ever. It's never Patty, right? St. Patrick's Day, that's grand. That's fine. No problem. Paddy's Day, sure, that's okay. St. Pat's if you have to, but not Patty, ever, right? And in the immortal words of my fellow Irishman, Liam Neeson, you know, if you continue to call it St. Paddy's Day, I don't know who you are, but I will find you and I will kill you, okay? It's definitely St. Paddy's Day. Okay, so now that we've gotten the important business out of the way, let me start talking about what I'm actually here to talk to you guys about today, which of course is antibiotics and antibiotic resistance. And so before I get started, I want to do a little exercise. I want everybody in this room to put your hand up if at any point in your life you have taken antibiotics. Right. I'm pretty sure that's almost everybody in the room. Keep your hands up. Keep your hands up, okay? Now I want you to keep your hand up if you've been on antibiotics twice or more. So basically put your hands down if you've only had antibiotics once in your life, but keep your hand up otherwise. Okay, still pretty much everybody. What about 5 times? You've been on antibiotics 5 times or more? Most hands are still up, right? I know I have. What about 10 times? Think about it. You think you've been on antibiotics 10 times? We still have a lot of hands up, right? I know I have, and many, many people have been on antibiotics multiple times. Okay, you can put your hands down. To those of you who've been on antibiotics once or more times, did you ever stop to think what might have happened if there were no antibiotics when you caught that infection? And particularly for people who have been on antibiotics multiple times, 5 times or 10 times. Did you ever stop to think, out of all those 10 times that you were on antibiotics, if there were no antibiotics here, would you be here now if it weren't for them? And there's certainly, I would say, a few people in this room, and it's probably the case, that you might not be with us today if it were not for antibiotics. And so it's sort of a sobering thought to think that if we didn't have these drugs, if they weren't available to us, that there's a number of people in this room that probably would not be here. And so it's kind of scary. And that brings me to the topic that I want to talk to you about today, and that is this growing rise of antibiotic resistance. Because we are now facing a real, genuine threat. Okay, we have antibiotics and we have developed them and they have been available, but we are facing a future where antibiotics will not work anymore. And so you think back to those times that you've been on antibiotics and the times that they've actually cured infections. If we now have antibiotics that are no longer effective, how is this going to work for us in the future? And are we now going to be susceptible to infections and susceptible to dying from infections that in the past were treatable? And as I say, this is a big problem. It's a growing problem. And here's an example of some of the numbers of infections and deaths that we're now seeing in the United States that are caused explicitly by antibiotic-resistant bacteria. And so this was from a report that was made by the Centers for Disease Control in 2013. And the numbers are estimated at about 2 million illnesses annually, 2 million illnesses every year in the United States alone caused by antibiotic-resistant bacteria. Now, these are bacteria that previously were susceptible to antibiotics but are no longer susceptible. So 2 million infections annually, which results in about 23,000 deaths. 23,000 people die in this country every year from antibiotic-resistant bacteria. Now, these numbers do not factor in the numbers of infections and deaths caused by antibiotic-sensitive bacteria. Remember, we're just talking about resistant strains here. These are big, big numbers, and these numbers are getting bigger. To try and put these in sort of a context that you can wrap your head around, if we're talking about 2 million illnesses every year in the United States, that's the population of New Mexico. So an equivalent population of the entire population of New Mexico are contracting illnesses by antibiotic-resistant strains of bacteria every year, resulting in 23,000 deaths. Well, 23,000, that's the population of Athens, Ohio. So an equivalent of the entire population of this town is dying every year in the United States because of antibiotic-resistant infections. So this is a big problem, and it's getting a lot bigger. All right, now that I've thoroughly scared you, I can go on and start to tell you what my talk's going to be about today. So I'm going to break it down into 3 sections. I'm going to start, discuss the history of antibiotics, what they are, where they came from. And then I'll talk about the rise of these superbugs. Where does antibiotic resistance come from and how it's spreading? And then finally, we'll try and lighten the mood a little bit towards the end and talk about some of the things that we're doing, as I say, in my lab to try and combat this, and some of the things that you can do, that general members of the public can do to try and combat this. So the first thing I'm going to talk about are these antibiotics. So the first question is, what is an antibiotic? Okay? I think when many of us think about antibiotics, we think, I'm sick, I'm lying in bed, I'm going to take an antibiotic, it's going to cure me, and bam, I'm back on top of the world again. And this is, functionally what antibiotics do. But literally, what are they? What are these pills? What are these chemicals that we're taking? And here's a definition of what an antibiotic actually is. An antibiotic is a substance, it's usually a chemical, that's produced by a microorganism. Antibiotics are made by microorganisms. They're made primarily by bacteria and fungi. So it's a chemical produced by a microorganism that at low concentrations can inhibit or kill other microorganisms, okay? And I think there's a common misconception about this. These are not compounds that are generated in a lab. For the most part, these are actually organic molecules that are produced by microorganisms that have the ability to kill other microbes. And we can see this very, very clearly in the lab if you look at an experiment just like this. So here we have a mold growing, and this mold is producing a compound, and it's secreting it. and that compound's an antibiotic. And also on this same agar plate, we have an E. coli bacteria, and you can see that around this mold, where this antibiotic is being produced, this E. coli can't grow. And so we get a nice zone of clearing around our mold where this antibiotic is present, where it's either killing or inhibiting the growth of the E. coli. So again, just really emphasizing the point that antibiotics are produced by microorganisms. Well, that sort of leads us to the question, why on earth are microbes producing compounds that can kill other microbes? What's the point of this, and why are they doing it? Well, to start to understand this, we have to think about life on this planet. And so we now know that life on this planet is about 4.6 billion. We know the origin of the Earth is about 4.6 billion years ago. And bacteria have been living on Earth for almost approximately 4 billion years, a little under 4 billion years. I'm going to go with 4 because it's easy. So bacteria have been here for 4 billion years. We've only just turned up, right? We're newcomers. We just got here. But for the first 2 billion years that bacteria were on the Earth, they were alone. There was nobody else here. It was just bacteria. And for the subsequent 2 billion years, that there have been other organisms on the planet, including the arrival of mammals and stuff much, much later on. But for 2 billion years, bacteria were here on their own. For 2 billion years, they were here with other organisms. And all of the time that they've been on this Earth, 4 billion years, they've been competing with each other. And bacteria have been very successful. They've spread out. They've covered the entire surface of the globe. We find them in every single ecological niche on the planet. But in doing so, they're constantly in competition with each other. And it's this competition with one another for limited nutrients that has led to the development of antibiotics by bacteria. And so let me just try and explain this in simplistic terms. Let's, for example, take a sample of soil. Soil is teeming with microbes. There's bacteria, there's all kinds of microbes living in soil. And so in my simple diagram, if we look in our soil, we have all these different species of bacteria that are living there. And they're all competing with each other in that soil for limited nutrients and limited resources, right? There's only so many nutrients and resources available in that soil. And so we get competition. And to try and outcompete some of its competitors, one particular species of bacteria, for example, this blue one down here, is going to evolve a strategy where it's going to produce a compound, and that compound, once it releases it from the cell, is going to kill its neighbors. And in doing so, it's cleared an ecological niche for it to survive in. And so now it can claim all of those nutrients that are around it, and it can go on and it can multiply and it can occupy that niche. And so this is a really good strategy, right? You kill off your competitors, you then can occupy that space. And this is really where we feel the origin of antibiotics has come from. And so this has been going on, as I say, for billions and billions of years. And then something happened not too long ago, about 70 years ago, which I've tried to depict by this red arrow up here. Something happened that's kind of changed all of this. And of course, what happened is us. And specifically, about 70 years ago, a Scottish scientist, Scottish, not Irish, Alexander Fleming, in 1928, he discovered penicillin. And then penicillin eventually went on to be used to fight bacterial infections beginning in the early 1940s. And we all know this, where everybody here is familiar with penicillin. It's really hard to overstate how much of an impact penicillin made when it was first introduced. Particularly in World War II, a lot of battlefield injuries resulted in infections, patients were septic, and penicillin was incredibly powerful and incredibly useful in treating these infections. And so it led to the kinds of posters that you see here, thanks to penicillin, he will come home. And so a lot of these traumatic battlefield injuries were now treatable, were now curable. Here we see another one of the uses of penicillin, but I'm not going to go into that today. These were incredibly powerful drugs when they were first developed. Penicillin was the first one, but it wasn't the only one. And this timeline here sort of shows us the golden age of antibiotic discovery. And so starting from around the 1940s, we see the development of these antibiotics. Running into the 50s, 60s, and 70s, and this is what we refer to as this golden era, or this golden age of antibiotic discovery, where it seemed like every month a new class of antibiotics were being discovered. And you can see many of them depicted on this graph here, and some of these you'll be familiar with the names. Well, how were these being discovered? Well, they were being discovered in probably the most simplistic manner you can imagine. Scientists were going out into their backyard, scooping up samples of soil, bringing it into the lab, seeing what microbes they could grow, and whether or not they secreted a compound that could be used to kill other microbes, and if they did, they would develop it as a new antibiotic. And this was incredibly efficient. All of these classes of antibiotics, pretty much every class of antibiotics that we still currently use in clinical practice were discovered this way, simply by digging bacteria out of the dirt and seeing if they produced an antibiotic. So really, we didn't develop these. We just found the bacteria that were already producing them, and we harnessed them, and that's how we developed antibiotics for use. So that's what antibiotics are, and that's really where they came from. So that's the first part of the talk. The next thing I want to talk to you guys about, of course, is the development of resistance. Question?
So I don't know anything about this, but I was wondering if you could explain the process of how microbes kill other microbes.
Do you mean the mechanisms of action of the antibiotics? Okay, well, I didn't want to go into too much detail on that today, and there's no one simple answer to that. The question was, how do these antibiotics work? How do they work to kill another bacteria? And so there's no simple answer to that. Antibiotics have different mechanisms of action or different ways that they do it. By and large, there are 4 major ways that most antibiotics work. The first one is destabilizing or destroying the cell wall. So all cells have a cell wall or a cell membrane. If you disrupt that, you kill the cell. So that's the first major mechanism that antibiotics use to work. The second mechanism is protein production. So inside the cell, inside the bacterial cell, inside the eukaryotic cell, we have ribosomes which produce protein. If you can inhibit the function of a ribosome, you can kill the cell. So that's a second major mechanism that antibiotics use. A third major mechanism involves DNA and RNA. If you can disrupt the DNA in the cell, if you can prevent that DNA from replicating or being transcribed into RNA, again, you'll kill the cell. So that's a third major pathway. And then there's a fourth pathway, which is a little bit obscure, which is folic acid biosynthesis. Folic acid biosynthetic pathway is targeted by a number of antimicrobials as well. So those are the 4 major pathways, but there are variations, and every antibiotic has a slightly different way of functioning, but those are the major ways. Is that okay? All right. So those are our antibiotics, and it's wonderful. We've got this huge repertoire. We've got this huge bank of antibiotics that we can use. But obviously, the next part of my talk, what I want to talk about, is the rise of these superbugs and where this resistance is coming from. So let's go back to our timeline where we have all these wonderful antibiotics to choose from. And, you know, people kind of got a little bit giddy. We were literally developing antibiotics every week. It's like, okay, I mean, this is fantastic. We can use all these antibiotics to treat all these diseases. And it led to this famous quotation from William Stewart, who was the US Surgeon General in 1967. And the quote that's attributed to William Stewart is, "The time has come to close the book on infectious diseases. We have basically wiped out infection in the United States." And so in the mid-60s, there were so many new antibiotics, new classes of antibiotics, so many being developed, we really started to feel that, you know, infectious diseases, we don't need to worry about these anymore. We can now forget about infectious diseases and go on and worry about other things like cancers or heart disease, things like this. This is often used as a really good example of speaking a little too early. Because as we all now know, we have not basically closed the book on infectious diseases. And the reason for that primarily has been the rise of antibiotic-resistant bacteria. So what am I talking about? Antibiotic resistance. What are antibiotic-resistant bacteria? So to put it in simplistic terms, if we have our bacteria, here's our normal bacteria over here, and we apply an antibiotic, which is this yellow, that bacteria will die. However, in certain cases, we can have the development of an antibiotic-resistant bacteria. And this bacteria is not killed by that antibiotic. As is depicted here, he survives in the presence of the antibiotic and then can go on to replicate and produce offspring. And so if this was during an infection, if you were infected with this antibiotic-resistant strain and you take an antibiotic, that antibiotic won't kill it. And that strain can continue to replicate and continue to cause disease in your body. And so antibiotic-resistant bacteria have the ability to survive in the presence of an antibiotic that would normally kill them. Well, the next question is, where is this coming from? Where is antibiotic resistance coming from, and how is it being developed? Well, to start to address that, we need to go back and look at our timeline of planet Earth again. And so we've already talked about how bacteria have been here for about 4 billion years. And for 4 billion years, they've been competing with each other. But we also know that in biological terms, every action has an equal and opposite reaction, right? And so bacteria have started to find ways to survive in competition with other bacteria. And it works a little bit like this. If I go back to my simplistic diagram of my blue bacteria is producing this compound, he's secreting it, killing those that are surrounding him. Well, the other bacteria in the soil, they're not going to be okay with this, right? All life wants to survive. And so bacteria have evolved and have developed ways to resist being killed by those antibiotics. And so if they can develop a resistance mechanism, it then allows them to survive in the soil also. And so for billions and billions of years, we've had this sort of constant battle going on in the soil and in all the environments on Earth between these different groups of bacteria. Okay? And I like to kind of oversimplify it on a little chart that looks like this. And so if we look at a timeline down here for about 4 billion years ago up until kind of present day, antibiotic use, the amount of antibiotic use, has remained, let's say, somewhat consistent, right? We have bacteria that are living in the soil that are producing these antibiotics, okay? And then we sort of have an equivalent level of antibiotic resistance, okay? Wherever we have antibiotics being developed, we're ultimately going to get other strains of bacteria that are going to evolve ways to survive in the presence of these antibiotics. And so for billions of years on Earth, we could imagine a scenario where this antibiotic use and antibiotic resistance have kind of gone hand in hand in this sort of parallel manner. Well, now we also know that about 70 years ago, something happened to change all this. What happened 70 years ago? Huh? Penicillin, right. So 70 years ago, Alexander Fleming discovered penicillin and we started using it. And so the amount of antibiotic use on this planet skyrocketed about 70 years ago compared to what had been used before. We now mass produce antibiotics in giant vats and sell them and use them. And so the amount of antibiotics being used on the planet absolutely took off astronomically about 70 years ago. Now, I'm not going to give out any prizes, but can anybody guess what's going to happen to the red line immediately following this astronomical rise? We're going to see the same thing, all right? And that's exactly what we see happening, is that we've started to use antibiotics much, much more than they've ever been used before on this planet, and we're now starting to see, with a very, very short lag time in between, we're starting to see this rapid increase in antibiotic resistance on the planet as well. Now, how exactly is this happening, and what's really fueling this rapid rise in antibiotic resistance? We know why there's an increase in antibiotic use, because we're doing it and we're making it. But why is there such an astronomical rise in antibiotic resistance, and how is that happening? Well, to try and explain that, I got to explain, first of all, what vertical gene transfer is. Vertical gene transfer is how pretty much all organisms pass along their genes. It's how everybody in this room acquired their genetic material from your parents. Okay? Your parents pass along their genetic material to their offspring, and that's vertical gene transfer. Okay? That's how most organisms pass it along. And that's how bacteria do, too. If you have a bacterial cell, it divides and it passes on its genetic material to the two daughters. And so that's vertical gene transfer. But bacteria have an ability that we don't have. And that's an ability that we term horizontal gene transfer. And let me explain to you how horizontal gene transfer works. As the name suggests, this is the ability to pass DNA from one fully formed cell to another cell. It's not passing DNA from a parent to a daughter cell. It's passing DNA between two fully formed cells. And so in simplistic terms, this is how we can look at horizontal gene transfer. If we have two bacterial cells here, one of them is resistant to penicillin, and the other is sensitive to penicillin. It can be killed by it. Somewhere on the genome of this bacteria, it has a gene that allows it to be resistant to penicillin. And so the presence of that gene is what's conferring this ability. Well, bacteria have this ability to horizontally transfer genes. And what that means is this cell can replicate some of its DNA and pass it horizontally to another cell. And this cell can acquire that DNA, and if that piece of DNA happens to have that gene for penicillin resistance, well then now this cell becomes resistant to penicillin. Yes, question? So the question was, does horizontal gene transfer occur on a random basis, or is there a mechanism for it? The answer to that is there are a number of mechanisms that drive horizontal gene transfer. And I didn't go into the specifics of how a lot of these work. One very common mechanism for this is viruses. So there are a whole range of viruses that can infect bacteria. And those viruses can infect a bacteria, take some genes out of that bacteria, and move them into a different bacteria. And so that's a very common mechanism of horizontal gene transfer, but there are others. And it happens with incredible frequency. It happens all the time, right? There's been some numbers that have been estimated of the number of horizontal gene transfer events that occur in the oceans on a daily basis, and it's something like 10 to the 23 horizontal gene transfers every day. So this happens a lot, and this is a really powerful mechanism that's driving the spread of antibiotic resistance, is the ability of bacteria to transfer these genes horizontally. Yes? Yeah. So the question is, does this only happen between bacteria of the same species, or can it happen intra-species, between two different species? The answer is both. It can happen between bacteria of the same species with maybe a slightly higher rate, but it can also happen between bacteria from completely different species. Yeah. And some bacteria are better at it than others, but yes, it can happen across the board. Yeah.
Sorry to take you backwards just a little bit.
It's okay.
Was Fleming, was he specifically looking for and discovering all that path, or was it truly an accident?
Yeah, the anecdote goes that somebody left an agar plate on the bench and went away for the weekend and came back and this mold had grown, and that he noticed, much like the picture that I showed, that there was this zone around the mold where his bacteria hadn't grown. So the anecdote goes somebody messed up and came in on Monday morning to go and throw everything in the trash and start again and noticed this. That's, like a lot of scientific discoveries, serendipity played a role. Whether or not that's true, but that's the anecdote. Yeah. Any other questions? Yes?
Bacteria are transferring segments of DNA as we
That's correct, yeah. So the question is bacteria transferring segments of DNA and that's being integrated into the genome of another strain? Yeah, that's exactly what's happening, yes. No, it's not. Again, there are a few different types of mechanisms. I wouldn't have time to go through them all today. In fact, if you want to take my pathogenic bacteriology class, I'll definitely talk about them. But yeah, they're not necessarily replacing genes. Bacteria can take foreign DNA and just integrate it on their genome and not have to replace it. They can just find a spot and put it in and keep it as a new piece of DNA. That's one mechanism, but there are a variety. Yeah. Yeah. So is the question, are certain people in certain age ranges more susceptible to infection by antibiotic-resistant bacteria? Yeah, that for sure. So there are, neonates, so young children, the elderly, people with suppressed immune systems. I don't know if there's a lot of evidence to suggest that the more times you take antibiotics, the more susceptible you are. I have never seen any studies like that, but who knows? I mean, now that we understand a lot more about the human microbiome, about how many bacteria there are living in us and on us, now we're starting to understand how much taking antibiotics can affect that and how much that can leave us susceptible to other infections. I'll talk a little bit about that towards the end. But I'm not aware of any studies that know exactly how more susceptible you are if you had your handle for 2 or if you had your handle for 10. Yeah. Okay? Any other questions? Okay. So this is our horizontal gene transfer, and this is one of these major driving events in the spread of antibiotic resistance. And so how does this work? How does by horizontally transferring DNA, how is that really resulting in an increase in these antibiotic-resistant bacteria? And to think about that, let's just look really simplistically at how antibiotics work, right? So here we have a population of bacteria right here. Maybe this is an infection. Maybe you've got a throat infection, okay? We're going to use an antibiotic, which is this pinkish color, and the presence of that antibiotic should destroy those bacteria, and then your infection has been cleared, okay? However, if one of those bacteria manages to become resistant, all the antibiotic is going to do is it's going to eliminate all the non-resistant bacteria. And it's going to leave that one there. And now that one bacteria can go ahead and replicate and reproduce. And so what we find is the use of antibiotics, if there's an antibiotic-resistant bacteria present, the use of antibiotics, all that does is it eliminates all of the sensitive bacteria and it selects for the resistant ones. And so the more antibiotics we use, the more we are eliminating sensitive bacteria and we are encouraging the resistant bacteria to develop and to spread. And that's another major driving factor behind the emergence and the spread of these antibiotic-resistant strains. So we know the bacteria are really good at developing this resistance and passing it on to one another. And that's really what's behind this sharp rise in the resistance that I mentioned. I want to take a minute just to really talk about how quickly bacteria can adapt. We're humans, we evolve a little bit slower, but the bacteria are really, really good at this. And to highlight this, what I'm going to show on this graph is on the top, I'm going to show the approximate date when an antibiotic was developed and started to be used in clinical practice. And along the bottom, I'm going to show you the date when the first reported case of resistance emerged to that particular antibiotic, okay? So for example, here we have penicillin. So penicillin, as I said, started to be used clinically in the early 1940s. First case of antibiotic resistant, the first case of a bacteria that was resistant to penicillin emerged about 2 years later, okay? So very, very short timeline. Here's tetracycline. Many of you probably heard of tetracycline. We see a very, very similar story. We see the introduction of tetracycline in the mid-40s and the development of resistance by 1950. And what we see is this pattern is repeated again and again and again. Every time we introduce an antibiotic and begin to use it in clinical practice, usually between 2 to 5 years, the first case of antibiotic resistance emerges. Hand up. So viruses are a whole other story. You're talking about bacteria? Sorry, so repeat the question. Well, it's that horizontal transfer that I mentioned, right? And so the ability to pass an antibiotic resistance gene from one bacteria to another, that's what's really driving this, okay? So if you think to the scenario where in the soil you have a bacteria producing an antibiotic and you have a soil bacteria that develops resistance to that. And this happened billions of years ago, okay? Well, maybe that was a resistance gene to penicillin. And so this soil bacteria that's resistant to penicillin can take that gene and can transfer it to Staph aureus. And now you have a penicillin-resistant Staph aureus. And that's what's really driving this. These antibiotic resistance genes exist in nature. They exist in bacteria in the soil. And by our overuse of antibiotics, we're essentially forcing these bacteria that infect humans to find a way to acquire those resistance genes. And once they do, they then spread out and become the natural population. Does that make sense? Okay, yeah. So I think, why does using antibiotics lead to the development of resistance? Okay. Well, not necessarily. You have to remember that there's maybe a limited number of bacteria that actually infect humans. Maybe there's 100. But antibiotic resistance, in many cases, these antibiotic resistance genes evolved in, say, for example, soil bacteria. Soil bacteria that would not necessarily infect a human. And that gene has passed somehow from this soil bacteria to a bacteria that does cause disease in humans. And so when that bacteria, this human disease-causing bacteria, causes an infection, and we use an antibiotic, you're selecting for it. You maybe kill off any other members of that pathogenic family that don't have that resistance gene, and you allow the ones that do to survive. And if you don't clear the infection entirely, and I'm going to get to the whole finish your course of antibiotics at the very end, but if you don't do that and you do leave some strains alive, those are the ones that then take over. And if you spread that infection, then anybody who gets that infection downstream is always going to have a resistant bacteria. Does that make sense? Yeah. Yeah, that's a great question. So once an antibiotic-resistant bacteria emerges, do scientists go in and tweak the antibiotic to make it work again? Yes, the answer is yes. And for about 20 years, that's all we've been doing. All we've been doing is taking all of those antibiotics that were developed in the 60s and 70s and modifying them and making them work again temporarily. I think the cephalosporins are a great example. We're on our 5th generation of cephalosporins. So we just keep modifying them to make them work a little bit more, but ultimately the bacteria catch up within a couple of years. Maybe not stronger, but they function again. You know, they get around the resistance mechanism, at least temporarily, but again, ultimately the bacteria seem to catch up. And that kind of leads me to an interesting point. To those of you who are medicinal chemists in the audience, will probably have spotted that a couple of the antibiotics that are listed on this slide are actually completely synthetic. Okay, so I told you that most of the antibiotics that we have are made by microbes, and that the reason that resistance has developed is because another microbe in the soil has developed resistance to that. Well, a couple of these, linezolid and daptomycin, are 100% synthetic. These are compounds that were created in a lab. And there was a lot of hope for these compounds because people thought, well, if these have not existed in nature, if there's no microbe out there in the soil that produces these, well, then no microbes in the soil have ever been exposed to them. And so microbes have had billions of years to develop resistance to these guys, but they've never seen these guys before. So these ones are going to work, and they're going to work for a lot longer. Well, you can see how successful that was. Bacteria have always developed and have always evolved ways around any drug that we've ever developed. Okay, so that's the scenario with our antibiotics and how quickly bacteria can evolve resistance to. But, you know, there's millions of bacteria in the soil, right? And so we just go out, we dig up more soil, we come in the lab, and we find more antibiotics, right? Just like we did in the 60s and 70s, right? We got an arsenal of these things. We just keep making more. These ones run out, we just make more. Well, unfortunately, that's not what's been happening. And this graph depicts the number of new antibacterial agents that have been approved by the FDA over the last 20 years. And as you can see, there has been a very, very sharp decline in the number of new antibiotics developed. Between 2008 and 2011, only 2 new antimicrobials were developed and brought onto the market. And one of the main reasons behind this has been that every major pharmaceutical company has now discontinued research and development of antibiotics. Well, this might come as a bit of a shock, and it's a huge problem. But if you think about it from financial terms, there's absolutely zero incentive for a pharmaceutical company to continue research in antimicrobials. It takes about $50 million to develop any new drug, to get it through clinical trials. So if you're the CEO of a pharmaceutical company, you've got $50 million to invest. Are you going to invest it in, number one, a new antimicrobial? And if it's successful, when it goes on the market, you've got 10 years of patent protection, because you've got 10 years to use it before Walmart starts making their version. And every time it's prescribed to somebody, they'll take it for 2 weeks, so maybe 10, 12 pills, and then they stop. Or, are you going to invest your $50 million in a high blood pressure medication? Whenever it's prescribed to a patient, they'll take that drug every day for the rest of their lives. Which one are you going to develop? It's a no-brainer. And all of the pharmaceutical companies agree. And so they've discontinued research into antimicrobials, and they're now really focusing on drugs where they can get people to take these drugs every day, which of course is the direct opposite of what we want people to do with antibiotics. And so as a result of that, we're seeing less and less development. And it's really falling to academic institutions, universities, people like me and microbiologists that work at universities to kind of carry the torch now and to continue this research into antimicrobials. Because as you can see from the talk today, these are badly needed, and there are some strains of bacteria that there are very, very limited options left when it comes to treating them. And here they are. This is the rogues gallery. This is taken from the CDC's Antibiotic Resistance Threat Reports from 2013. And this lists the most serious threats in the United States right now posed by antibiotic-resistant bacteria. They're divided into 3 categories: urgent threats, serious threats, and concerning threats. I'm not going to go into the reasons as to why these are in different categories, but some of the bacteria that are listed on this board, there are no antibiotics that are currently effective in treating them. And that is an incredibly scary prospect. I'm going to talk a little bit later on about Methicillin-Resistant Staph Aureus, because that's the bacteria that I work on. But we see down the bottom here, there's a vancomycin-resistant Staphylococcus aureus, or a VRSA. It's only down here in the 3rd level of the concerning threats. Vancomycin-resistant Staphylococcus aureus, there are no antibiotics that can treat this. An infection from a VRSA strain is completely untreatable. Thankfully, there have only been 14 cases in the United States since 2007. So it's extraordinarily rare, and it's never gotten past the first patient. But if a strain like this were to get out and cause an epidemic, it would be very, very serious. Okay, so these are the bugs that account for these 2 million illnesses and 23,000 deaths. Yes, question?
Wait, so if
It's because it's rare. Yeah. When you think about the number of 2 million infections, there's only been 17 of these. 14. That's the only reason it's down that low, because it seems to be very rare and it just hasn't really kicked off, if you like. Not like some of these, that are a lot more common. So that's part of the reason these are in different categories is simply the frequency of the infection. Yeah. What exactly does it infect? It infects us, humans. Yeah, it's, you don't have to be very scared because, like I said, there's only been about 14 cases of it. But it's similar in most respects to Mrsa, and it infects in the same way. And I'll talk about Mrsa infection. So the same would be true of VRSA as well. Yeah. Yes, question.
Where in the United or where in the world were these 14 cases? Were they in America or in
The 14 cases were in the United States. Yeah. Yeah. Any other questions? Yeah. Are most of our present antibiotics synthetic? No, most of our present antibiotics are natural products. Now, they may have been modified somewhat from the original form produced by the bacteria in the soil, but they're based off of a scaffold or a compound that was produced naturally by a bacteria. There are very few examples of synthetic antibiotics. Yeah. Yes?
So I've heard that my doctors, finish your antibiotics and whatever you have to become resistant to antibiotics. Is that true? And if so, how does that happen if they're in resistance naturally occurs when they're crossing these species?
Yeah, so the question is, I've been told by my doctor to always continue finish the dose. You know what, I actually have a slide on that at the end, so is it okay if I just answer that question when I get to the end? Yeah, okay. There was another question here?
Sorry, thinking back to horizontal transfer.
Sure, I love horizontal gene transfer.
I'm having a hard time understanding why bacteria would utilize interspecific horizontal transfer because if they produce these antibiotics to capitalize on a niche and outcompete other microbes, why would they take the chance of transferring this resistance
Yeah.
to other species?
Well, the answer to that is it's not necessarily always the bacteria's choice. Oh, so the question is, in horizontal gene transfer, if a bacteria in the soil has evolved and developed an antibiotic, why would it be beneficial for it to give that antibiotic to a different strain? That's the question? Yeah, so it's not necessarily always the bacteria's choice, right? So if you're a bacteria and you're infected by a virus, that virus kills you, takes your genome, and that can then package it up and move it on to a different strain. So there's other factors at play. Yeah. Yes, question. Not sure I follow the question. Can you repeat it? Right. Do the antibiotics themselves evolve over time? There will be slightly different versions, for sure, yeah, and they may have different efficacies, again, against different types of organisms, right? So one antibiotic doesn't always kill every other type of microbe, right? So there are specificities involved, and there are variations. There's not just one penicillin that's produced by one fungi that is active, right? There's varieties and there's different forms of it. So yes, you do see different variations and different forms of the antibiotics from different species, yeah. Okay? Yep. Can a person take multiple antibiotics at once? Yeah, and for many diseases, and some of the ones that are actually listed on the board here, you would be prescribed multiple antibiotics to treat it. I think the top one is multi-drug resistant TB. Multi-drug resistant tuberculosis, if you're diagnosed with it, you'll take 4 antibiotics for 6 months, and then another 2 antibiotics for another 6 months. That's how hard it is to clear multi-drug resistant TB. Yes? Yeah, that's a great question. If a species of bacteria develops resistance to an antibiotic and then that antibiotic is discontinued or it's not used a lot, will they eventually lose the resistance to that? And there's some evidence to suggest that that does happen. But to try and utilize that as an effective strategy, we would have to synchronize every doctor on the planet and say, right, everybody, on Tuesday, we stop using tetracycline. And 10 years' time, maybe we can use tetracycline again. So there is some evidence that they can lose it over time, but whether or not that's going to make a difference for us in the long-term strategy here, it's probably not going to be beneficial. It's not going to help us. Yeah. Okay. So, moving on to the last part of my talk, unless there were any other questions. No? Okay. What can we do? What are the new approaches, and what can you guys do to try and combat this? So, you've seen this. I put it up a couple of times so far in my talk. The number of infections, 2 million, and about 23,000 deaths caused by antibiotic-resistant bacteria. Well, there's one character on that list that is responsible for the majority of these deaths, and we've already mentioned it, and that is Methicillin-Resistant Staphylococcus aureus, or MRSA, or Mrsa for short. Methicillin-resistant Staph aureus causes about 80,000 infections in the United States every year, and it's responsible for a little over 11,000 deaths. So almost half of these deaths from antibiotic-resistant bacteria are specifically due to this one bacteria here. And that is the bacteria that I study in my lab, so I'm just going to take this opportunity to talk to you guys a little bit about Mrsa and sort of some of the ongoing projects that we have in our research lab that are trying to combat this issue or address this issue of multi-drug resistant bacteria, and particularly methicillin-resistant Staph aureus. So, first question I always get asked when I tell people that I work on Staph or Mrsa is, what is the difference? What is the difference between Mrsa and Staph? So here's my ridiculously simplified diagram to show you this. If this green circle represents all of the strains of Staphylococcus aureus that we know of, the red circle represents methicillin-resistant Staph aureus. It's a subset of that group. And so Staphylococcus aureus, or Staph for short, so you talk to somebody and say you got a Staph infection, it's a Staphylococcus aureus infection, right? And MRSA is a subset of that. Now Mrsa, or Staph in general, is a really interesting bug. So there's a lot of bacterial pathogens that cause one specific disease. Think about something like cholera, right? If you ingest cholera, it gets into your intestines and it causes disease in your intestines and you get diarrhea and you get sick. That's what cholera does. And many bacteria are like that. They cause one specific disease. Mrsa is not like that. Mrsa is an incredibly versatile pathogen. It's versatile in terms of the locations in the body that it can infect, but also in the severity of the diseases that it can cause. And let me just highlight this with some of these slides. So this slide here, Mr. Skeleton, shows you some of the locations that Mrsa can infect. Mrsa can infect the brain, can cause meningitis and brain abscesses. It can infect the lung and cause pneumonia, the heart, causing endocarditis. It can cause food poisoning, toxic shock syndrome. Mrsa really loves infecting bones and bone joints, so it causes septic arthritis and osteomyelitis. And you guys have probably heard that diabetic patients are very susceptible to amputation. The primary reason for diabetic amputations is Staph aureus osteomyelitis infections, okay? So these are a variety of different locations that Staph aureus can infect. It's not just one location. But of course, on this slide, I don't have the most common organ in the human body that's infected by Staph, which is the skin, okay? The majority of Staph aureus infections are in the skin, but it also has the ability to get inside our bodies and cause infections in all of these different locations. Once it does that, the severity of the infections there's a wide range. On the one hand, pun intended, it can cause these relatively mild infections. Things like soft tissue infections, boils, and skin abscesses, things that look like this, okay? But then a step up from that, it causes relatively moderate infections, things like impetigo, cellulitis, and scalded skin syndrome, which you can see this patient suffering from here. And so it looks like these are burns and the skin is peeling off. That's actually not the case. This is an infection. It's a Staph aureus infection, and a toxin that's produced by Staph aureus causes the skin to peel off like this. If you haven't had your dinner yet, look away, because now we're going to talk about the severe infections that are caused by Staph. Things like toxic shock syndrome, septicemia, and what's depicted in this picture here, which is necrotizing fasciitis. Necrotizing fasciitis, aka the flesh-eating disease. It is never a flesh-eating virus. We just put that one to bed as well. This is where the fascial planes become degraded, the action of the bacteria. As you can imagine, these diseases have a high rate of morbidity and mortality associated with them. Septicemia, Staph aureus in the bloodstream, without antibiotic treatment is 90% fatal. Now, thankfully, the frequency of infection goes inverse to severity. And so these severe infections happen relatively rarely, whereas the most common infections are these relatively mild skin infections up top. And probably most people in this room have had a staph infection in the skin at some point in their lives. So what is it about staph that means it can cause all this variety of diseases in all these different locations in the body? Well, unlike a lot of bacteria that might produce one toxin or maybe two toxins, staph produces a lot of toxins. So just depicted on this cartoon slide behind me are 30 different toxins that are produced by Staph aureus, that can be produced by Staph aureus. And this isn't even it. Right? This is just an example of 30 different toxins that can be produced by Staph aureus strains. And this goes some way to explaining why it's such a versatile pathogen and why it can cause so many diseases in so many parts of the human body. So we have a bacteria that's becoming increasingly resistant to antibiotics. It's a little bit different because it produces a lot of different toxins. So maybe designing a vaccine against one of these toxins, that's not going to work, right? And that's true. We don't have a staph vaccine. And part of the reason is because it produces so many toxins. So what can we do? How can we develop new ways to combat infections by staph? Well, one of the approaches that we're taking in my lab is to look at all of these different toxins and try and find some commonalities between how the bacteria makes these toxins. And one mechanism that we're looking at is how the bacteria makes these toxins and pumps them out of the cell. Okay, so this is supposed to depict the inside of the bacterial cell, and here's the outside. And so all of these toxins are made inside the bacterial cell, and it's got to pump them out of the cell so that they can go on and then attack your body. Now, when it does this, all of these toxins get pumped out of the bacterial cell using the same system. And so all of these different toxins have to go through the same pathway in order to get out of the cell. Now that to me represents a very, very narrow bottleneck. And if we can design some way to inhibit this pump, if we can prevent this pump from working, then all of these toxins are going to be blocked from exiting the staph cell. Okay? And so that's one of the things that we are now working on in my research group. And so the idea here is can we design a new drug that will stop the bacteria making these toxins? And if we do that, we're not going to kill the bacteria, right? The bacteria is going to survive, but it's not going to be able to make those toxins. And it's the action of those toxins that makes you sick and kills you. And so we like to refer to this strategy as the disarm but don't harm approach to developing new therapies, to developing antivirulence drugs, which will prevent the bacteria from being dangerous, but won't necessarily kill them. And so we've had some success from our preliminary studies. Here is an example of looking at the activity of one particular toxin that's produced by Mrsa, okay? And so here we have the bacteria here. This is our normal Mrsa. And what you're seeing here, this black zone that's surrounding the colony, this is the action of one toxin. So this toxin is being made, it's being pumped out of the cell, and the amount of black that you see around here, that's showing you the activity of this toxin. Well, this is a strain of staph that we've been able to construct in our lab. And as you can quite clearly see when you compare the two, there's much less activity of the toxin in this strain. And so this is one of the approaches that we've taken. And so what we'd like to do now is to try and develop a compound, a drug that might inhibit this pump, which we could then apply to this strain and prevent it from producing these toxins. And so that's one of the things that we're doing right now. But most of you don't work in research labs. Some of you do. I think I saw one or two people that work in my lab here. But most of you don't work in research labs, and so you're not going to be able to work on projects like this. So the question is, what can I do? Is there anything I can do to help combat these antibiotic resistance bacteria? So let me go through a few things. The first thing is the simplest, and that is to wash your hands. I know in flu season you always see these wash your hands slides, and it's true. Washing your hands with soap and water, there really is no substitute for this. A lot of people always ask me, oh, what about antibiotic hand soaps and, you know, these antibiotic or hand purification things? I say, you know, they're good in small doses, but you got to be careful because there's been a lot of reports lately that a lot of these antibacterial hand soaps are really not as effective as they're marketed as being. Here's an example of an article from Time magazine where a bunch of these hand sanitizers were told by the FDA to stop advertising themselves as being effective against Mrsa because they are not. And here's another research article from a colleague of mine who showed that triclosan, which is a common antibacterial in soaps, hand soaps, and toothpaste, actually promotes colonization of Staph aureus. So the presence of triclosan can actually increase the colonization of staph in your body. And this is used commonly as an antibacterial in soaps. So I tell people there's no substitute for soap and water and washing your hands. The second thing you can do is only take antibiotics when you need them. Knowing the difference between a cold, which is not treated, can't be treated by antibiotics, and an actual bacterial infection is critical. And so part of the blame comes on us. We shouldn't go to the doctor and be, oh, I need an antibiotic, right? And part of the responsibility is also with the physicians, who shouldn't overprescribe antibiotics just in case it's a bacterial infection. And so knowing when we need an antibiotic and only taking it when we need it is another critical step. The third thing, which we already talked about, is finishing the full course of your antibiotics. And this is absolutely critical. And so part of the reason for that is if you only take your antibiotic and you kill off the bacteria that are susceptible to that antibiotic, and then you stop taking it, well, all you leave behind are the bacteria that were resistant. Okay? And then this becomes the predominant population. But the other reason why it's very important to always finish your course of antibiotics, if you remember way back to the early slides, I told you that antibiotics either kill or inhibit another microorganism. And so there's two ways that antibiotics work. The first way is that the antibiotic directly kills the bacteria. And so in this example, we have our bacteria, we add an antibiotic, and they're killed. Once we discontinue that antibiotic, the bacteria are gone, and so there's no problem. But there's a second class of antibiotics that don't directly kill the bacteria themselves. All they do is slow down their growth. And they slow down their growth long enough for our immune systems to come in and clear that infection. And in this case, the bacteria are not directly killed by the antibiotic, they're just slowed down for a long enough period to allow our immune systems to work. If you don't finish that course of antibiotics, there's a chance that you don't give your immune system enough time to clear that infection, and once you remove the antibiotic too soon, there's enough bacteria remaining that they can go on and reproduce and continue the infection. And so there's a number of reasons why it's critical that you always finish a course of antibiotics. Take a probiotic. There's been a lot of research into probiotics recently, and we now understand a lot more about our gut health and how much our good bacteria play a critical role in that. There is a whole series of infections and a whole series of deaths that are caused in the United States every year by people taking antibiotics and resulting in an infection because of a bacteria that can take hold after you take antibiotics. And the idea of using a probiotic is a way that we can combat this. And the bacteria in question is Clostridium difficile, or C. diff. Now, C. diff is a little bit different than the bacteria I've talked about so far. It hasn't acquired resistance. The thing with C. diff is it's always been resistant. Most of our antibiotics, got to wrap it up? Okay. I got like one more slide, okay? So what happens if we take an antibiotic, we know that our natural bacteria in our gut, if we take an antibiotic, we kill those off. And what that does is it allows bacteria like C. diff an opportunity to grow because it's naturally resistant. And so when we use an antibiotic, it can allow harmful bacteria to grow in our gut. And taking a probiotic to supplement our intestinal bacteria is a way to combat that. And so it's always a good idea if you're on antibiotics to take a probiotic at the same time. And the final thing I wanted to talk about is supporting antibiotic-free agriculture. This may come as a surprise to you to know that only 20% of antibiotics used in the United States are used for humans. 80% of all antibiotic use in this country is agricultural and is used for livestock. Okay, that's fine. We all eat meat, so we want to keep our cattle healthy, right? So it's okay to use these antibiotics when our cattle get sick. Well, that's not what we're using them for. Only 14% of those antibiotics are used on sick animals. 86% are used in a non-therapeutic dose. Why is that? Well, a couple of years ago, it emerged that if you give livestock a low dose of antibiotics, it increases the weight of the animal by 10%. So if you've got a herd of livestock, you give them all a low dose of antibiotics, it increases the weight of your herd by 10% and increases the amount of money that you get by selling that herd by 10%. And so the use of antibiotics in livestock and in agriculture in the US is absolutely rampant and results in about 80% of all antibiotic use. This practice is banned in Europe. You're not allowed to use antibiotics in livestock in Europe. And it's a practice that we really need to stamp out in this country. It's massively contributing to the rise and spread of antibiotic-resistant bacteria. Okay, that's it. I realize I've gone a little bit over time. Thank you all for staying. I'll answer any more questions. Yeah, absolutely. The disarm, don't harm is very similar to the antibiotics that don't kill the bacteria, right? Slow them down enough that you'll allow the immune system to clear the infection. Yeah. What's the most dangerous bacteria? The one that kills you? I mean, it just depends on the person. It depends how your immune system is. It depends if you've been exposed to it before. It varies. One of the worst is Acinetobacter baumannii. It's got incredible levels of resistance. Staph aureus kills a lot of people. Yeah. Yes.
Fear that when you actually develop or stop the hospital, fear that they would develop resistance to that,
Well,
and
the
like chasing it.
yeah, it's of course it's a possibility because as we know, bacteria will develop resistance to anything. But the idea that if you don't kill them, it puts much less pressure on them to evolve ways around that. So we talk about selective pressure from evolutionary terms. And so if you're not killing the bacteria, it's thought that that puts less evolutionary pressure on them to mutate, and perhaps it will slow down the process. But ultimately, yeah, they'll ultimately probably find a way around it.